Today I am going to present a paper which is Indigenous Probioticlactobacilli in combating bacterial infection and insights. Before going to the main part of the presentation, some part is there to focus for clarification as introductory. So first of this, the theme is probiotic. That is why you have to know what is probiotic. Probiotics are live microorganisms belonging to the group of lactic acid bacteria and mainly the lactobacilli, the genus Lactobacillus. There are many species. These are live microorganisms, and when we consume those, these provide health benefits and different ways. And so, in probiotic research, this is widely distributed throughout the world, and locally they show different effects. So we have tried to isolate some locally available sources, the lactobacilli, the lactic bacteria, which can be used as probiotic. Here we used cord and stool sample from infants. Thereafter, we have identified and performed safety profiling and the antibacterial activity, probiotic features, and their bacteriocin from as a antibacterial agent. Because such kind of bacteria lives three types of mainly the antibacterial agents, which are hydrogen peroxide, lactic acid, and bacteriocin. We have studied here the bacteriocin, and now just we have to see how these can these isolates, lactobacillus isolates, can be isolated from different sources. These can be isolated from any kind of fermented foods or milk or infant stools, but we have utilized two samples here, cord and infant stool. First, we have inoculated MRS broth, which is Demon Rogosa Sarpi broth, and followed for growth at 37 degrees Celsius temperature in MRS media. So when we see the lactobacilli growth, we have just streaked on the plate of same media, including agar. That means MRS agar media. We have followed the streaking plate method for the isolation of single discrete colony to get pure culture of lactobacilli. So after getting the pure culture, we have used one test which is known as TSI, triple sugar iron. That means in this test we can see whether H2S is production is taking place or not, and three sugars, triple sugars, that's why glucose, lactose, and sucrose are fermented or not. Then we proceed for gram staining, spore staining. These are the main characteristics tests to define the lab bacteria. That means probiotic lactobacilli. Then we followed oxidase test and catalase test. When such bacteria found as gram positive rod, that means bacilli shaped, and this is spore non-forming, catalase negative, oxidase negative, then these are lactic acid bacteria, and this is rod shape. That's why this is lactobacilli, lactic acid bacteria under which it belongs, and the genus is Lactobacillus. And thereafter we followed for tests to get the identity as species, and that's why you have performed carbohydrate test. We utilized 20 carbohydrates for this test, different biochemical test, and 16S RNA ribotyping. RNA gene for ribotyping was sequencing. We use different lactobacilli. The table is showing that feature, and after that we have submitted the sequence to NCBI gene bank. And in the last column, their number are showing in the form of which the isolates are deposited. And thereafter we have blast at gene bank website to get the main identity of the lactobacillus. As for examples of so many, we have isolated here presented Lactobacillus fermentum, and this is the phylogenetic tree of a isolated bacteria. Thereafter, Lactobacillus plantarum. This is a card isolate also. And the third one, which is represented here, the matched is Lactobacillus fermentum. And our isolate is Lactobacillus fermentum also, which is designated as LMEM 10, LMEM 15, and LMEM 22 isolates. And within the bracket, the NCBI number are recorded. So now we have to see how these are probiotics. Why we will call those probiotic? That means we have to justify or authenticate the probiotic features of isolated lactic acid bacteria. So we performed different tests such as physiological stressor tolerance test, pH, because a probiotic lactic acid bacteria, when you consume, we first uh, see that these bacteria face in our buccal cavity with the saliva where lysozyme is present. We have seen that lysozyme resistance is showing this bacteria up to 10 milligram per ml. This is one criteria of probiotic which are matching with our isolates. And I am back with the previous slide. pH tolerance, low pH. Suppose we have taken the isolate, we consume orally, and then it reaches to the stomach. In stomach, HCl is secreting, and there is due to acid production, the media is acidic. That's why we have tested for low pH tolerance test, and thereafter the bacteria migrate to the intestine for their colonization and showing the health benefit differently. And there the range of bile salt extend up to 0.5 percent, and at this level our isolates have shown resistance, and they have shown their growth. Initially the growth was 5.699, and in this table we are seeing that in different hours their number were increased with the presence of different stressors. But other kind of bacteria did not exist in this type of condition, but these are growing well under in media in vitro system to confirm as these are probiotics. So these are first batch of strains, 4 strains: Lactobacillus animalis, Lactobacillus plantarum, Lactobacillus acidophilus, and rhamnosus. And in the last panel, Lactobacillus kg, Lactobacillus fermentum, and the middle column there is single strain Lactobacillus fermentum. And we have thereafter authenticated that these are not pathogenic. Therefore, we have tested different tests such as hemolysis. 3 kinds of hemolysis are exhibited by bacteria, which is alpha hemolysis. In this hemolysis, there are greenish zone are formed around the colony. In beta hemolysis, transparent zone are found, which is shown in this slide. And in gamma hemolysis, there is no zone. That means due to non-hemolytic nature, these lactobacilli are non-pathogenic, and these are not producing any zone, greenish or the clear zone. And thereafter we performed, these are not showing here, thereafter we performed gelatinase activity. That means gelatin liquefaction is occurring or not, or DNA test. We see that our isolates all were non-pathogenic because these were gelatinase negative and DNA is negative. But other control positive strains such as Staphylococcus aureus, Proteus vulgaris, these are positive for the test, but these are pathogenic. Next, in red color, blue color, and green color, we have isolated from 3 sources, respectively from homemade commercial card, homemade card, and infant stools. And these all were shows similar character. These are non-pathogenic and very stressor tolerance to sodium chloride, bile salt, and low pH. After that, we have tested their antibiogram. That means antibiotic susceptibility test of lactobacilli, because a isolate of lactobacillus intended to be used as probiotics must be sensitive to antibiotics. But antibiotic resistance is also considered when these resistance are not transferable. We have seen that mostly our isolates showed different types of antibiotics, but some strains shows resistance to some antibiotics, such as ciprofloxacin. See this table here, according to MIC by agar dilution method, and this is Dix diffusion method. By agar dilution method as well as Dix diffusion, some isolates show resistance to amikacin, gentamicin, vancomycin, and methicillin. These are actually proved as non-transferable. So our isolates were authenticated as good probiotic isolated from our locally available food sample and stool sample. Now we have tested for antibacterial activity of such probiotics. Before that, we have discussed about the indicator strains which are pathogenic and gram-positive 3 strains: Bacillus cereus, Enterococcus faecalis, Listeria monocytogenes, and gram-negative Acinetobacter baumannii, Escherichia coli, Klebsiella pneumoniae, Pseudomonas aeruginosa, Salmonella typhi. This is gram-positive. Somehow this is shifted here. This Staphylococcus aureus is gram-positive, Proteus vulgaris, and Escherichia coli. We have used such this kind of indicator bacteria against which we have seen that such Lactobacilli shows antibacterial resistance, and such bacteria show different types of resistance pattern. We have tested 16 antibiotic, and these column indicate their resistance number of antibiotic, and this divided by this. That means last column divided by the last before column. That means RA divided by number of antibiotic indicate the MAR indices. When MAR indices is more than 2.2, then these are originated from a source which is highly antibiotic contaminated. And when the value is greater than 0.4, such as Pseudomonas aeruginosa, see this 0.625. Here is Proteus vulgaris 0.686, and another isolate also is these isolates shows more than 0.4 MR index values, and these are probably originated from stool contaminated, human stool contaminated sources. And now the antagonistic activity, that means the antibacterial activity of Lactobacilli against the indicator bacteria that we have shown earlier. Here we have followed the susceptibility test by agar overlay method. In this method, the Lactobacilli were grown on spot, that means by spot culture on MRS agar plate. Then after 48 hours, we overlay nutrient broth culture and nutrient agar, semi-liquid nutrient agar mixed with the indicator strain and pour over it, and then dry it, solidified, and incubate. And thereafter, next day, we have seen that this spot, due to the presence of Lactobacilli, shows the zone of inhibition. They have acted on the indicator strain, different types of, such as Acinetobacter baumannii. These A, B, C, D, these are different types of lactic acid bacteria, Lactobacilli, and these act against Acinetobacter baumannii, E. coli, Proteus vulgaris, Salmonella typhi. And the inhibition zone around the spot culture of Lactobacilli indicate their antibacterial activity by agar overlay method. Similarly, we have followed agar well diffusion method. In this method, we have swabbed, inoculated on the surface of the Muller-Hinton agar media. Then we cooked well and pour the culture filtered of the Lactobacilli, and we have seen after their growth at 37 degrees Celsius ambient temperature at atmospheric carbon dioxide in presence of, and we have seen that in this method the Lactobacilli showed excellent antibacterial activity, showing the zone of inhibition around the well. And from the agar overlay method we determine R, and from the agar well method we determine bacteriocin activity in the form of arbitrary unit per ml, which is zone diameter of inhibition into 1000 divided by volume taken of culture filtered, and R is the inhibition of this zone diameter minus the diameter of this spot lactobacilli divided into 2. This is R. The R value and the AU per ml value are represented in this table respectively, and these are very high. That's why they are showing very excellent antibacterial activity against all the microorganisms. That means gram negative and gram positive bacteria were noticed. Here, to conclude about their nature of probiotic property, we have determined or calculated cumulative probiotic potential in 5-point score. We have seen that our isolates were acid resistant, bile salt resistant, showed antagonistic activity, antibiotic sensitivity, and non-hemolytic. And so we have seen that in 0.54 scoring, except one, all other strains showed 100% CPP. That means got the score 5, except one, which is LMEM, and this got the 80% score. But in our panel, we have seen that more than 80% CP value indicate the good probability of the Lactobacillus isolated. One thing is very important, which is bacteriocin, because I have already told that these bacteria shows antibacterial activity due to different kinds of antibacterial agents, among which bacteriocin is important, very important. That's why you have isolated bacteriocin from all isolates. Here is the representative. This is SDS page panel. We have used and marker protein, and this is our isolates bacteriocin, and we have seen that this is 13.1 kD Dalton molecular size. This bacteriocin, when tested by agar-well diffusion method against Acinetobacter baumannii, gram negative and gram positive Bacillus cereus, both are potential pathogen. They showed very good antibacterial activity because of the presence of very large size inhibitory zone. Here are the tables. We have determined the zone of inhibition for different pathogenic bacteria by using this bacteriocin, and determine also the MIC, minimum inhibitory concentration of bacteriocin. And this feature shows that these are, that means bacteriocin isolated from lactic acidobacteria are excellent antibacterial agent. And we have lastly authenticated, we know that bacteriocin are low molecular weight protein or peptides. Okay, so we have seen whether it is protein or not. In our case, we have seen that control strain shows 21 millimeter zone diameter against E. coli and Acinetobacter baumannii 23. These are 2 gram negative bacteria and 2 gram positive bacteria. These are pathogenic Staphylococcus aureus and Listeria monocytogenes. Against this, without any treatment, the zone diameter were 24 and 17 millimeter respectively. But when you treated with trypsin and proteinase K, then their activity were significantly lost. But when amylase were treated, their activity were not significantly lost. That means proteinase and trypsin activity is hampering the activity of bacteriocin. That means degrading the bacteriocin. That's why these are proteinaceous nature in our case of isolates. Thus, we have seen that the locally available sources are very good source to isolate lactobacillus, and lactobacillus are good probiotics because of their different types of stress tolerance, such as sodium chloride, bile salt, temperature variation, and these are non-pathogenic because of non-hemolytic, DNase negative, and gelatinase negative, as well as they are having no resistance antibiotic property, transferable resistance property, as well as these are mostly sensitive, and these are showing both our isolates as well as the bacteriocin isolated from such isolates as very good antibacterial agents. So these are fulfilling the all criteria in vitro of probiotics, and that's why we can be used as good non-antibiotic biotherapeutic agents, at least in our part of the world. So, remaining part maybe we have to test for pharmacokinetic study because of dose determination and further toxicity testing in modeling system or in clinical trial. And thereafter we may get circulated for commercial use. But locally we may use in the form of card because we have isolated such bacteria from card, locally available card. And these are my publications in the field of lactobacilli, Probioticlactobacilli. And finally, thank you. And this is acknowledged to my PhD scholar, Devashish Haldar, for who is performing this type of study in my laboratory of Gour Banga University. And as well as I acknowledge Department of Science and Technology India for grant and sequencing for Krumas Biotech Private Limited India. Thank you very much. Thank you.